Good evening. My name is Navya,
And my name is Saranveer Kaur.
and today we're going to discuss our floating leaf test experiment, photosynthesis in kale and spinach.
To begin with, first we need to clarify what exactly is photosynthesis and what we need during this process. So to carry out photosynthesis, plant need carbon dioxide, water, and light energy. And in this experiment, we use kale and spinach to explore the effect of different intensity of light energy on the rate of photosynthesis. In order to do so, we will be measuring the amount of oxygen produced, which is a byproduct of photosynthesis, and the number and rate of floating disk determine the rate of production of oxygen, which in turn is a direct measure of the rate of photosynthesis. The rate of photosynthesis vary from higher rate in the leaf disk exposed to bright light to no amount of photosynthesis occurred in dark.
Okay, so to begin with, as we know, plants use light energy and carbon dioxide from the atmosphere, along with water and nutrients from the soil to create their own food. This process of conversion of light energy into chemical energy is known as photosynthesis. Here is the chemical equation of photosynthesis. This is accomplished by leaves using specialized pigments known as chlorophyll. The process by which plants containing chlorophyll convert carbon dioxide and water into carbohydrates using light energy is called photosynthesis. In this experiment, the effect of light energy on the rate of photosynthesis is measured using two species of leaves, namely kale and spinach, by monitoring the sunken leaves placed in different intensities of light. Okay, so our thesis statement for this project is that if the intensity of light energy is taken as one of the important factor affecting the process of photosynthesis, then it is predicted that the rate of photosynthesis is supposed to be higher in bright light than in room light.
Now comes to the method section. Fresh leaves of kale and spinach were used and 90 leaf discs were cut. Using a syringe filled with bicarbonate and soap solution, 10 leaf discs were added at a time in the syringe and vacuum was removed from the leaf discs. The rate of photosynthesis is measured by observing the number of leaf discs at a regular interval of time. These vacuum leaves will then be transferred to the cups containing 40 milliliter bicarbonate soap solution. The rate of photosynthesis is measured by observing the number of leaf discs at a regular interval of time. The bubble produced around the leaf surfaces are the indication of the production of oxygen. This oxygen is responsible for the buoyant force produced inside the leaves.
For the control of our experiment, we used distilled water with soap solution instead of bicarbonate solution to create a solution without carbon as a source of photosynthesis. So this is a picture showing leaf discs, sunken leaf discs. Air from the leaves is taken. So we used a bright bulb as a source of our light. 3 cups were placed near a bright light source and the readings were taken for the amount of time a leaf disc took to reach the surface of the cup. Mean readings were recorded and compared when the experiment was repeated. So this is an image showing the floating of a leaf disc. Here we can also see the production of air bubbles, which is actually oxygen. So for our results section, we found out that for both spinach and kale, only the leaf discs floating were observed in bright light conditions. The leaf discs did not float in room light during the course of 20 minutes, as represented in our table. Also, the leaves kept in dark did not float because of the absence of light as a primary factor necessary for photosynthesis. Spinach leaves and kale leaves display almost similar floating trends, with spinach showing faster photosynthetic rates because the leaves were floating on the surface quickly.
And here is the graph derived from the result. At the time zero, none of the leaf discs were floating in either of the cups. And this graph is for the bright light, just because none of the leaves were floating in dark and the room light. As the time passes, after every 2 minutes of interval, leaf discs were found to be floating. And at the course of 10 minutes, all of the leaf discs in all of the cups were found to be floating. And the standard deviation after all the calculations comes out to be 3.125, and the mean value of the floating discs is 6.08. And here's the table representing the mean data of floating leaf days of spinach and kale in different light conditions from the conducted experiments: bright light, room light, and dark.
It was observed that in the bright light, leaves started floating, whereas in cups placed in room light, dark, and control group displayed no change. The possible reason for these observations can be that in the dark there was no source of light to carry out photosynthesis, hence no change was observed. Whereas in room light, leaves did not get enough light to excite the electrons for the initiation of the light reaction process, which is responsible for the synthesis of oxygen. Therefore, the rate of photosynthesis is slowed down. These placed in bright light floated at a much higher rate. Therefore, increasing the intensity of light will increase the rate of photosynthesis. Therefore, the result it coincides with our hypothesis. So, before going to conclusions, there are many variables which may have affected our results in during the experiment.
For instance, lack of bicarbonate ions in the bicarbonate of solution, human errors in reading for time intervals, vacuum leaves not placed inside the cups containing bicarbonate solution at the same time. Some of the leaf discs could have veiny parts, leaf discs not vacuumed properly or overly vacuumed, faulty syringes with back vacuum used.
So, for our conclusion, through this experiment, it can be concluded that to carry out photosynthesis, the appropriate amount of light energy or source of light is necessary. Both spinach and kale leaves display similar floating trends. All leaf discs were floating on the surface in 16 to 18 minutes. Since the leaves did not float in room light and dark conditions, this means that the intensity of light plays a major role in the process of photosynthesis and in turn oxygen production. Thank you.